Very good morning to all the delegates. We are moving on to the next talk by Dr. Debasish Bandopadhyay on cardioprotective efficacy of melatonin as observed through in vivo and in vitro mechanism based evidences in experimental studies. For this talk, we would like to invite our two chairpersons, Dr. Neelambige ma'am. Presently, Madam is working as Dean and Professor of Physiology at KVG Medical College, Sulia, Karnataka. Madam did her MBBS from Tanjore Medical College, MD from Madras Medical College, and MBA in Hospital Administration at Alagappa University. And she's ex-Senate member of MGR University, Secretary Ethics Committee. And Madam has many achievements and awards to her credit. She had received Best Doctor Award from District Collector of Coimbatore, has received Women Achiever Award from Jettle Book of Records. She has conducted several UG and PG assessments throughout India. And she is presently member of Academic Board of JSS University and Kerala Institute of Health Sciences. Has worked as Medical Council of India Nodal Officer at Government Medical College, Coimbatore. Has served as Dean at Kanchipuram Medical College, Enathur, Tamil Nadu. And presently, Madam is working at Karnataka, and she's my mentor, teacher. I'm very happy to invite you, ma'am.
Next, I'd like to welcome Dr. Omlata Bhagat, the Additional Professor of AIIMS Jodhpur. Ma'am has completed her MBBS from Gajaraja Medical College, Gwalior, and from her MD from Mahatma Gandhi Memorial Medical College, Madhya Pradesh, and her Masters in Community Health from Jawaharlal Nehru University, Delhi. Ma'am has rendered preclinical, paraclinical, and clinical services in various institutes. Her expertise is over autonomic, vascular, and pulmonary functions. She has the 31st Dr. S. Radhakrishnan Memorial National Teacher Award for 2015 for contributions as medical teacher, and her field of interest lies over cardiovascular physiology, especially cardiac autonomic responses to exercise, respiratory training, yoga, and meditation. Ma'am was also an associate editor of IJPP and has a number of publications of more than 18, and as a resource person has 2 chapters and 25 abstracts. She has been a regular resource person since 2011 in EPFICACY and ASOPICON. We welcome you, ma'am.
Over to chairpersons, you can introduce our
Thank
speaker.
you. Thanks for the invitation. For giving. I take privilege to introduce Dr. Bandopadhyay to the floor. He is the founder and head of the Department of Biotechnology in AIIMS, Assam, and he headed the Center of Biotechnology, Vishwabharati University. He did his PhD from CSIR, Indian Institute of Chemical Biology, Calcutta, and he has many professional achievements and awards. He received the prestigious Parimal Vikas Sen Memorial Award from Physiology Society of India, Calcutta, and he is working in the field of melatonin since last few decades and has made significant research contribution on melatonin in the domain of enzymology, oxidative stress, free radical biology, myocardial ischemia, gastric ulceration, which has been recognized world over. Moreover, he has also made an impact in the field of development of new drugs, antioxidants, and of sympathetic and natural origin. He has national and international collaboration and published more than 125 papers till now. Now I invite Professor Bandopadhyay to deliver his lecture. Please, sir.
Yes, very good morning. And thank you very much for introducing me so nicely. I first would like to thank the organizers, Chairman, Sir, Madam, and other people responsible for the conference. And I feel really honored to be invited here because, you know, I have accepted this invitation more than 2 months or 2.5 months ago. But suddenly I have to move out from Calcutta. So I am now talking to you from outside Calcutta, and hopefully the internet facilities will be all right and electrical facilities will be all right during the lecture. So I would like to present slide. I would like to share with you all. I hope you can view me, see me, and also can have my voice also.
Yes, sir, we can hear you well, sir.
Okay, thank you very much. As you can see, today's talk is about cardioprotective efficacy of melatonin as observed through in vivo and in vitro studies and mechanism-based evidences in experimental studies. But I would like to inform you that melatonin is a natural molecule. At the very first point, I would like to mention that melatonin is synthesized and secreted from pineal gland located within our brain. And although the physiological level of melatonin is up to the order of picomole to micromoles per liter of blood.
Excuse me, sir. Sorry for the interruption, sir. Can we put the slides in slideshow, sir? Slideshow mode.
Yes, I'm not started the slideshow yet.
Okay, sir. Thank you.
Slides are working. Okay, sure. This is the introductory slide. Yes. Can you see the slides?
Yes, sir. Now we can see it in slideshow mode, sir. Thank you, sir.
Okay, sure. Definitely. So, but initially, people talked about third eyes in the brain and tried to relate it with pineal function and other things. But in 1998, melatonin, in 1953 to 58, this time period, the structure of melatonin was determined by Aaron B. Lerner. And after that, in 1998, melatonin was shown to be a very strong antioxidant molecule. And at the same time, in 1963, there was a paper in Nature whose title was the scientists could not find an LD50 for melatonin. That was the first indication that even if melatonin is administered or consumed through food or through supplementation from microgram to gram quantities, the people may not be affected. That means the molecule has no side effects. That is one of its biggest plus point as an antioxidant, because a lot of antioxidants, they may be synthetic, they may be natural. As soon as they react with toxic species, that may be reactive oxygen species, reactive nitrogen species, chlorine species, or other drug metabolites or xenobiotics, as soon as they react with the molecule, they become converted into pro-oxidants. So antioxidants becoming converted to pro-oxidants is a more dangerous situation for cellular health. So people were looking for decades for antioxidants whose end products will not be toxic, will not be having side effects. And in case of melatonin, it was found that the intermediary metabolites arising out of melatonin within the cell are even better scavenger molecules, even better antioxidant molecules compared to melatonin. So chances of melatonin getting toxic to the body when administered or even being accidentally consumed by any person is remote, is very rare. So with this background, when I started research works on melatonin years back, so this was the background which prompted me and my laboratory students to continue research on melatonin. So let us have some details about ischemic heart disease and acute myocardial infarction that are currently major health threats and have reached unprecedented levels, every one of us know, in the urban as well as the rural society. Particularly in India, the myocardial episodes in rural population was almost nil, maybe 30 years back even. But nowadays, many patients come, even farmers having ischemic problem. This is because only due to the changes in economic and dietary scenario, their lifestyle changes, and all these are the things that almost every one of us know. So reactive oxygen species are toxic molecules that we know. These are oxygen metabolites. You add one electron, it becomes superoxide, anion, free radical. You add another one electron, it becomes hydrogen peroxide. You add one more electron, it will become hydroxyl radical. And you add one more electron, it becomes water. So, coming to a stable state, but the intermediate stages are very, very different and very difficult to handle with. So reactive oxygen species are toxic molecules, intermediates of oxygen metabolites, which are generated as a result of reduced blood flow to the heart and are thought to be responsible for the generation of oxidative stress. Even many hundreds and thousands of diseases, people have found oxidative stress as one of the major components. Sometimes, drugs even don't work, because if you apply drugs which get transformed into toxic metabolites within the body and the body itself gets stressed, then how body can handle these intermediate compounds. That's why, many of the drugs fails. Instead of providing benefits to the patients, they fail to become effective. Mitigation of such oxidative stress induced myocardial injury leads to improved cardiac performance. So isoproterenol is a chemical compound that is a synthetic catecholamine as well as a beta-adrenergic receptor agonist, has previously been reported to produce gross and microscopic infarct in rat heart, which are similar to that produced in human. Melatonin, as I was talking about, a pineal secretory product, has been shown to possess antioxidant properties and protects the cardiac tissue and shields against the harmful effects of oxidative damage. Now it is very well known that melatonin is effective as a supplemented material or as a drug or as an external antioxidant molecule to control, even in America, when lakhs of people were dying out of COVID-19, high doses of intravenous injection of melatonin saved many lives, many lakhs of lives in US, and many papers are also coming out. Many papers have been published regarding the efficacy of melatonin. So melatonin, a pineal secretory product, has been shown to possess antioxidant properties and protects the cardiac tissue, and that is the reason that it shields against the harmful effects of oxidative damage. So with this background, we have carried out a series of experiments to explore the protective effect of melatonin against isoproterenol bitartrate-induced rat myocardial injury in vivo and in vitro, and to elucidate the underlying mechanism of protection offered by melatonin against such damage. So what was our first question? that is there any oxidative stress generation? If there is oxidative stress generation in the rat myocardium, there must be generation of hydroxyl radical in vivo. So we first measured the hydroxyl radical formation in presence and absence of melatonin, and found that melatonin effectively is capable or effectively reduces the hydroxyl radical generation within the rat myocardium. But the question was not stopping here. Our next question was whether it provides protection against generation of superoxide anion. So we have measured xanthine oxidase, xanthine dehydrogenase activities, and calculated their ratios and summation, and found that melatonin effectively is capable of scavenging even superoxide anion-free radical, which is another reactive oxygen species within the rat myocardium. But if there is oxidative stress, there must be change in expression and activities of several of the proteins related to the defense mechanism of the cell, defense mechanism of the rat myocardium. So we have measured activities of copper, zinc, SOD activity, catalase activity. These are called housekeeping antioxidant enzymes, mitochondrial antioxidant enzyme, MNSOD, which is a very strong antioxidant enzyme, very strong protein. One cannot damage this protein this way, that way. So when this gets disturbed, that means the oxidative extent of oxidative stress is quite high, and glutathione peroxidase, entire peroxidase family gets disturbed when oxidative stress ensues. And we have also studied the expression of these proteins and found melatonin effectively protects against the changes brought about by isoproterenol. So these results we have published in a journal named Journal of Pineal Research. Thereafter, we switched our interest whether isoproterenol affects the energy metabolism within the rat myocardium. So we measured some of the Krebs cycle enzyme in vivo, like pyruvate dehydrogenase, which is the first enzyme, the entry pathway into the Krebs cycle, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinate dehydrogenase, and all of these enzymes you can find isoproterenol inhibiting the activity in vivo, and that's when pre-treated with melatonin with 10 milligram per kg body weight is the dose of melatonin we have used. We have carried out a dose response studies, and you go on increasing melatonin dose, you go on having the promoted or more effective activities of melatonin. But here we have carried out our experiments with the minimum effective dose of melatonin and tried to see what happens. So these results we have again published in Journal of Pineal Research in 2012. But following that, we have made questions to ourselves that what happens to the purified mitochondria. So we have isolated first the mitochondria from the goat heart, because in our earlier publications, we have shown that goat heart is a rich source of heavy mitochondria. And heavy mitochondria is a very good model for studies on oxidative stress. So we have procured goat heart from Calcutta Corporation approved slaughterhouse, isolated the mitochondria, purified them, and subjected to treatment with isoproterenol at the increasing concentration. And in another set of experiment, we have incubated the mitochondria with increasing doses, increasing concentrations of melatonin with a fixed concentration of isoproterenol that was found to be best effective. But the question was whether we were working with the live mitochondria. So we have checked with the mitochondrial viability, at the same time assess the activity of succinate dehydrogenase and mitochondrial potential. So these experiments have shown us that what you are working with live metabolically active mitochondria, that means respiring mitochondria. So we have studied the mitochondrial membrane lipid peroxidation and protein carbonyl contents concentration-wise, time-dependently, and found melatonin concentration-dependently protected these biomarkers of oxidative stress, indicating again that melatonin can serve as an antioxidant, can serve as a cardioprotective antioxidant in at least in rat model. Later on, if you can see this particular figure, I was talking to another plenary lecture here at the Bankura Christian College yesterday. This type of enzyme activity profile is perfectly showing that the oxidative stress is there. If MNSOD peroxidase activities are crisscross in your assay systems, then the oxidative stress situation is perfect, which is supported by E-redox measurement of that is mitochondrial membrane potential, which was found to be disturbed in isoproterenol treated rats or isoproterenol treated mitochondria, which was found to be also protected by melatonin. So again, showing that melatonin having strong antioxidant activity. So similarly, we have measured the in vitro hydroxyl radical formation and found that melatonin inhibits hydroxyl radical generation. Then we have found again the xanthine oxidase activity for superoxide and ion-free radical formation checking. Melatonin was also found to protect, and also is nitrite production, which got reduced when the mitochondria is subjected to isoproterenol in vitro. But melatonin provides protection and does not allow nitrite levels, because lowering nitrite level causes tremendous stress and alters the membrane potential. That is very much important. So in our experiments, melatonin protects membrane potential. At the same time, it prevents nitrite level to get reduced. So, but if this be the case, the next very question is that whether the proteins are oxidized while the stress is being there. So we have measured the dityrosine autofluorescence and tryptophan autofluorescence and found that yes, proteins are oxidized severely and melatonin is capable of providing protection to them. Similarly, but as I have shown in our in vivo experiments, that the Krebs cycle enzymes are inhibited. So the question was, what is the pattern of inhibition? Is there a common type of pattern they are exhibiting? Or the pattern may be different for different enzymes. So we have measured the activities of succinate dehydrogenase again in an in vitro system and found that there is a competition between isoproterenol and binding to the substrate binding site to the enzyme, and melatonin removes this competition through that means it is a competitive inhibition being removed by melatonin in its presence. Similar is the case with the isocitrate dehydrogenase. Earlier slide was pyruvate dehydrogenase, but this slide is isocitrate dehydrogenase, and you can see the competition between the isoproterenol and melatonin, isoproterenol and the active site of the enzyme with its own substrate, but in presence of melatonin, this competition is completely removed. So this is the same story with alpha-ketoglutarate, which is another redox enzyme of Krebs cycle. Similar is the case with succinate dehydrogenase, which is very important for mitochondrial metabolism, because activity enhancement and activity reduction have different meaning in mitochondrial oxidative stress language. So this inhibition of succinate dehydrogenase is removed in presence of melatonin in vitro. So then we have started some other people say that a depolarized mitochondria is shifted to left side of the graph, and so more a mitochondria is polarized, more is good, more good is its health. And if it is starting depolarization, then that means there are leaks within the membranes, It cannot hold the ions within inside, and the oxidative stress ensues in a very strong way. So, but people can ask question that you are talking about mitochondrial damage, but can you visualize the mitochondrial surface that to establish the fact that mitochondria is damaged not only from inside through the biochemical mechanisms, but morphologically it is also injured. So we have studied the scanning electron micrographic studies, and which shows in the third panel you can see the damaged and blebbed mitochondria, which was found to be protected from getting damaged when there is melatonin in the incubation medium. And we have pictographed the mitochondria in both bright field as well as scanning electron microscope, so as to establish that what we want to visualize is perfect in concept. So these type of data we have again published in Journal of Pineal Research in I think this is in 2015. So this is a third paper, Journal of Pineal Research from my lab in Calcutta University. So, but we continued our research. That the question was, is it only isoproterenol that damages heart, or the other oxidants can also have the same mechanism of damage, and melatonin can protect? Because many people asked me the questions in one seminar that if you can try with other oxidants and the same antioxidant. Okay, fine. So we have tried it in copper ascorbate model. We have isolated mitochondria from goat as well as from rat and incubated with copper ascorbic system, which is a very strong hydroxyl radical generating system, and found that melatonin can provide protection to this. But you can see, if you see the right panel of the graph, that means copper ascorbate can cause damage to the enzyme manganese SOD, but which is a mitochondrial enzyme, very strong antioxidant enzyme. And with increasing concentration of melatonin, you can have almost full protection. But if you perform a reversal experiment, the experiment gets failed. That means melatonin can protect the proteins from their activities getting declined, but it cannot reverse. That is a new information, that melatonin is a protective antioxidant, but it cannot reverse. So is the case with copper zinc SOD, which is classified as the housekeeping antioxidant enzymes. So we have also checked it with catalase also, but the story is the same, that reversal is not possible, and an uncompetitive type of inhibition was observed in case of pure, when if we use the pure enzyme, incubate with copper ascorbate and try to protect the enzyme or try to remove the inhibition with melatonin. So these are some of the experiments. With earlier experiments were with the enzymes isolated from the tissues, but these data that I am showing you are with the pure enzymes procured from Sigma Chemical Company. And this is of the catalase. That means that be it protein being coming from the extracted sources or protein coming from a purified source, the outcome doesn't change. This means that what we are observing is happening, and that is why the results are same and protections are same. So later on, we have tried to study the binding of melatonin with different type of toxicants in order to find out that apart from melatonin acting as an antioxidant scavenging the free radicals or toxicants, or working through MT1 or MT2 or maybe some hitherto unknown receptors through changing expression of enzyme proteins or other things, does it have some other mechanisms of action? So we tried to use, we have used pure enzyme like catalase and started its binding studies with copper ascorbate using isothermal calorimetry, and found that one site sequential binding with copper ascorbate. That means, by in genuine term of thermodynamic rules, the binding is happening, and that means catalyst can bind copper ascorbate and getting damaged. So we studied then catalyst with copper and melatonin, and copper with ascorbate, and copper with ascorbate and melatonin, and we found that you can see in the third panel that is sequential binding shows that melatonin has typically has a capacity to bind both the toxicant as well as the vulnerable sites of the protein molecule. Here the protein of interest is catalase, obviously, but it can happen to any protein. Catalase here is an antioxidant enzyme, but it can happen to a structural protein. It can happen to a signaling protein. It can happen to even a contractile protein like myosin or actin. That melatonin binds on one hand with toxicants, and on the other hand, it provides protection through binding to the vulnerable sites so that free radicals or toxic metabolites cannot have a place to bind and damage the protein, and accordingly the function of the protein. So people asked us that how can you confirm that the binding is happening? So we have used the pure enzyme with melatonin and found a regular one-side binding. A linear one-side binding clearly shows that the binding story that we were trying to establish is a truthful story. And the thermodynamic considerations have revealed that there is binding. Melatonin can bind not only at one site, but can bind with multiple sites. And what is the evolutionary purpose of this multiple site binding? It is only for protection, providing protection. Because then the literature came out. Many people are working, definitely. What we understand now, that chronobiological functions of melatonin man knew when the knowledge developed, science developed. But antioxidant function of melatonin is there right from diatom. That means melatonin evolved not as a hormone, but as an antioxidant. That means since 1993, the story got totally changed. That yes, it is a chronobiological regulatory hormone. Diurnal rhythm is regulated by melatonin. But these are knowledge came to human being later on. But it's what is the origin? The origin is of antioxidant function, because diatoms have been shown to have melatonin, which have structure not different from human melatonin. And this 2.5 billion of evolution to human being melatonin did not change its structure at any point of evolution. That is very important. So then we confirmed our studies with superoxide dismutase and using the same type of reaction mixtures as you have seen earlier, and we find a sequential binding pattern. That means this is not only happening with catalyst, this is happening with SOD also. And maybe at some different place in the world, someone may try it from myosin ATPase activity. Someone can try with some signaling proteins like SORT1 or any other proteins or whatever maybe, or some transcriptional proteins also. So I'm pretty sure that the results will be the same. Here also again we have confirmed that yes, melatonin binds separately with one site linear kinetics with superoxide dismutase. So these results we have published in Life Sciences recently. And so what is the take-home message? Melatonin's ability to provide protection against oxidative stress-induced myocardial injury is gradually getting well established. Isoproterenol-mediated competitive inhibition of mitochondrial enzymes and copper ascorbate-mediated alteration of activities of cardiac antioxidant enzymes can be significantly protected by melatonin Moreover, binding of melatonin with oxidants and vulnerable sites of enzymes provide new clue to its mechanism of action. This raises finally a great possibility of melatonin being considered for use in human alone or as an adjunct therapy in different cardiovascular situations and myocardial tissue injury. Here, at the end, I would like to repeat that melatonin saved millions of lives in the United States when factually and actually America was struggling among the world nations to control COVID-19. And this melatonin studies have been, treatment and studies both have been started in Texas first, and then, you know, in Massachusetts and other states of US, and they got exceedingly well success. And this was promoted in the Western world, even, you know, in doctors in Kolkata, and I hope in other cities in India, when they find COVID-19 patients hospitalized, they use nowadays high doses of melatonin. They use melatonin tablets, they use melatonin syrup, they use even melatonin spray to COVID-19 patients to reduce the cytokine storm. What is the special property of melatonin? To getting melatonin reduces cytokine storm and save lives of millions of people. So these are some of the funding sources. I must express my gratitude to support me and my scholars. And finally, thank you very much for inviting me to share some of our researches carried out maybe at least through 15 years of time. And thank you very much.
I congratulate Dr. Sir for your great research and perseverance and your contribution to the science.
Thank you very much.
It's a very great research and nicely presented. So I just want to have one simple question to you, that though you are doing very basic research in the area of melatonin and its cardioprotective nature, I just want to ask that what is your suggestion if you want to give to the clinician in prescribing the melatonin in the regular patients, not just COVID-19, in regular patients also we do have this.
My suggestion,
so I just want that.
Yes, my suggestion to the learned cardio physicians that they can now, the knowledge is at present that they can now prescribe 5 to 10 milligram melatonin tablets to the ischemic patients, I think on a regular basis at bedtime. And because melatonin does not induce sleep, it promotes sleep. So it will, on one hand, promote sleep. That means the ischemic patients need good sleep, quiet sleep, and at the same time will regenerate heart, at least regenerate the myocardial energy metabolism, apart from the other treatments that they are providing to their patients. So I think clinicians must not hesitate in prescribing melatonin, at least up to 5 to 10 milligram to patients. Okay, normal patients having some heart problems or hypertension problems, because in our laboratory we have shown that in hypertensive rats, melatonin is very much effective in reducing blood pressure to the normal level. So if blood pressure regulatory medicines are applied, are co-administered with say 5 or 10 milligram of melatonin daily, I think that will be wonderful for patients. And there are many patients I know, my friends and some of my relatives, I have suggested them to consume melatonin, are really keeping fine. Very good question.
Thank you, Dr. Bandopadhyay.
Most welcome, most welcome.
Yeah. Whenever we think of melatonin, we always remember a circadian rhythm and putting us to sleep. Today's lecture has opened a new concept as melatonin as a cardioprotective agent. So thank you very much for enlightening us with such an excellent lecture and your extensive research on the cardioprotective role of melatonin. I'm very happy that we are able to suggest to clinicians that melatonin really helps in cardiovascular diseases. So at the outset, I'd like to congratulate and thank Dr. Lakshmi Jatia and team for being a part of this third international conference on the role of basic medical sciences in academia, diagnosis, and research advancement. So I'm continuously, second time I'm in the screen. Thank you very much, team. And thank you very much, Dr. Omlata, for your input also. Best wishes to all of you.
Thank you, ma'am. We would like to thank our speaker and chairperson by showing our gratitude and token of appreciation by issuing a certificate and memento. Thank you, sir.
Most welcome, most welcome.
And we would like to thank our chairpersons also. Thank you, ma'am, Dr. Leelambike, ma'am, for chairing our session.
Thank you.
The next chairperson, Dr. Omlata Bhagat. Thank you, ma'am. Thank you for spending a valuable time. And thank you, everyone, for joining this session. And we'll move on to our next session.